So I presume there's a small typo in the agenda, or are you going to tell us about?
Yeah, I didn't study a whole year.
A whole year?
No.
That's an amazing talk.
That would be a lot longer than 5 minutes. So I'm just going to jump in with a little background here. You'll see from these numbers that the beluga hearing covers a much wider range of frequencies than humans, as well as a much higher range. And basically, sound waves enter through the jaw and that region and are transferred to the middle ear and then on into the inner ear. And here's where we find the cochlea. Now that's a small spiral-shaped structure which is fluid-filled and contains 3 ducts or channels, the central one of which contains what's known as the organ of Corti. If you'll notice the picture on the right, there are some purple structures there, and those are hair cells, of which there are 2 types, the inner hair cells, or IHC, and they form a single row. And then we also have the outer hair cells, or OHC, which are arranged in 3 rows. Overlying that is a layer of cells called the reticular lamina, and protruding through those are stereocilia, which are attached to the hair cells, and those are the structures we're going to be looking at most today. So you'll see here a flattened version of the cochlea if we uncoiled it and laid it out, and there are a number of morphometric and morphological changes that occur as we progress from the base all the way to the apex. And along with those morphological changes are changes in frequencies that are detected at those locations. So you can see things such as spacing of cells, the height of the stereocilia, and so on, all change. So why is this important? We know that the frequencies received in the cochlea at the base, higher frequencies are detected, and then as we move towards the apex, low frequencies are detected there. So if we can correlate those frequencies with their location, we can develop a frequency map for belugas. So for instance, if we found acoustic trauma in this region, we could potentially associate it with the sound that caused that. We can do that throughout the cochlea. So the basis for the methodology we were looking at is from a paper by Yaron, looking at guinea pig cochlea. And you might think, guinea pigs, they're tiny. Belugas are much, much larger. Where is the similarity? But there are many well-documented trends in mammals relating the morphological differences to the frequencies at any given point in the cochlea and how those change throughout the cochlea from the base to the apex. So the objective of the work I was looking at was to determine which of these morphometric features are frequency dependent within the inner ear of the beluga whale. Our samples were obtained through legal subsistence hunting, which allows for a fresher sample, less degradation, and then in turn, more reliable results in the end. So we started out using a scanning electron microscope to take micrographs of the cochlea and used ImageJ software to analyze those photographs to determine the measure of the cochlear length as well as morphological parameters. So we started taking the micrographs, and as we take each one, you have to tilt the sample to accommodate for the curvature of the cochlea. I looked at 120 photographs similar to this to measure out the area. Each one has to correspond to the next photo to ensure an accurate length. And our cochlea was about 38 millimeters in the end. So the next step was looking at 6 different locations around the cochlea to further analyze some of those parameters that I discussed. These are the 7 parameters that we analyzed. We did multiple measurements of each at each of those 6 locations, and we found trends in 4 of these that match the trends that we have well documented in terrestrial mammals. So you're going to see a few graphs, and just the horizontal axis is the distance from the apex. So we see we move from the apex to the base, and the y-axis here is, in this case, the parameter of the inner hair cell, that single row, all the way to that 3rd row of outer hair cells. So you can see here in the beluga, there's a decreasing trend. So as we move from the apex to the base, that distance decreases. And if we overlay the work from Yaron in the guinea pig, we see that there is a correlation among the frequencies. Now, on this graph, as well as subsequent ones, you're going to see that I have data points beyond the range of the guinea pig. So I'm going to touch on that later, but the guinea pig upper hearing limit is only 30 kilohertz, so that makes sense, but the trends are in agreement. So these are the other trends we saw. So again, the first graph we just looked at, and we also see a decrease between the gaps in that second row of cells, outer hair cells, and the third row of outer hair cells. So they all decrease as we move to the base. And then in the width of the outer hair cell, that actually increases. And these are all trends that are consistent in land mammals. And again, looking at the guinea pig, we've overlaid the frequencies. And again, as I touched on, there's the gaps, but I'm going to talk about that in a minute. So we feel that the current results are promising and preliminary, but show some potential. We want to do more research on numerous individuals, additional locations within the cochlea, as well as additional morphometrics. So just to move forward, where are we going to go from here as far as figuring out those gaps that I talked about? This is just showing you the frequency range for humans and then guinea pigs, which is a little more than ours. And then you can see the area of overlap between the guinea pig and the beluga. And then the full range of beluga hearing extends quite a bit further. So how are we going to figure out where those gaps are? We're going to look at echolocating bats. They have a similar hearing system as far as echolocation as in belugas, and they cover a similar hearing range. So potentially we can use that to fill in some of the gaps and make a full map. And this could also be a suitable alternative for traditional frequency mapping techniques. And I would like to acknowledge and thank these people without whom this research would not have been possible. Thank you.
Questions for Cassandra? You're okay with that. Thank you, Cassandra.